All right, so I guess reminders, there's another relatively minor concept check Wednesday. There's this mini project, which is the one you can't drop, that is due Sunday. It's basically implementing a very basic GA with analyzing. So you'll have a little bit of exposure to crossover, a little bit of exposure to mutation, and just generally solving this numerical optimization problem with the population. So just gives you some experience with that and kind of seeing what happens when you make different choices and how well the solutions come out. So hopefully that's helpful. Um, so today I wanted to just kind of wrap up then with the GA and answer some of the last questions that were brought up in the muddiest points, for example. So generically for any GA, it's going to have a structure along the lines of you start with an initial population, which normally we just think of as being randomly initialized throughout the decision space, but as with other optimization approaches, let's say training a neural network, for example, then there are different ways you can initialize your population. It doesn't have to be random. There could be reasons why you prefer sets of solutions that are in certain locations, for example. But for all intents and purposes, we'll just say randomly initialized. And then from there, you score the population using your fitness objective or your optimization objectives. So this is where your opt objectives come into play, like F, for example. So this is where you get your kind of trait values for each one of your, this is where you sort of see like, who's 5 foot tall, who's 6 foot tall, et cetera. So you could, if that helps, you can think of this as this is where you assess traits. Um, and then from scoring your populations, then you can check for termination. And so this, could be for, this could be number of generations. It could be based on convergence, and et cetera. So that's to decide, have we done this enough? And so if, for example, after you score these populations, you find out that the scores really haven't changed that much, at least the top scores, and it's maybe gone for 6 generations and things haven't changed that much, then in that case, maybe it's time to end. So this is potentially where you could, where you might end up leaving. So, you know, the, this, possible end here. And I should also mention that this is where your termination conditions will come into play. I guess I could keep track of all of the parameters. Your termination conditions come into here, your optimization objectives go into here, your population size goes into here. We'll call that M. And I guess you could also argue that their number of genes sort of is set up at this point as well. So I guess I could say, and number genes. So then if you've decided that you're not going to terminate, then at that point, and it's been subtly mentioned, but we're going to say that at this point we have an opportunity to scale the population. And so this is where any fitness scaling functions come into play. So you have your raw optimization objectives, your raw traits, but those don't necessarily map directly into fitness. And so you have an opportunity to squeeze certain differences, for example, so that wide differences in fitness maybe correspond to small differences in effective fitness that ends up going into the rest of the algorithm. So that's where you put that there, or you just pass things directly through. But so that is an opportunity for you to put in whatever your fitness scaling functions are. And from there, then you select parents. And so this is where your selection operators are introduced, or their parameters. And so if you use default selection operators, then maybe you've got parameters. So for example, this might be where you decide how many parents you want. So are parents. And so this, if there's any randomness in your selection, that's also where there's an opportunity for genetic drift to come into play. Because what randomness, generally when you say you just want to select your R parents, you're saying based on their fitness scores or their scaled fitness scores, we're going to decide who gets an opportunity to get into the next generation and potentially generate offspring, which are combinations of those. Well, if that was purely based on fitness, then there would be no opportunity for drift. But if 90% of the time a high fitness solution gets in, but 10% of the time a low fitness solution gets in, that, luck, so good luck for the low fitness solution, bad luck for the high fitness solution, that's where drift comes into play. So any selection operator that uses any bit of randomness provides the same opportunities that prevent you from washing out low fitness solutions, which maybe later actually might have some benefit as they recombine with others, whatever. That ability of a low fitness solution to hang on, or the ability of a high fitness solution to not be selected, that introduces the risk of losing solutions that you might want to keep around. And that's drift. So any randomness at the selection introduces the opportunity for drift. So then after you select your parents, then you select your offspring. And so this is where your crossover operator comes into play. And it's also where your, and I'll sort of draw this as maybe a separate dashed line. I'll call it the probability of crossover or a crossover fraction. So it is, and I'll draw this out for different algorithms, how this is implied, but this is sort of the fraction of parents that get an opportunity to recombine to create these mixture of offspring instead of just kind of clonal offspring. And so those are additional parameters that come into play. And then after that, then you decide who to mutate. So you have a mutation step. And so that's where your mutation operator, as well as the probability of mutation, come into play. And then after you've done that, you've selected good solutions, you've generated novel combinations and solutions. So we're ready to rescore and try it all again. So then it loops. So then you come back and you rescore your population and check for termination and start the whole process. Again, that is your generic GA. And you can view kind of this group that I've been down here as collecting kind of the hyperparameters. For the GA, so you have the opportunity to change these. You can swap them in. You can choose your own operators. You can choose your own values of this. If you go in to If you download a library in Python that implements the GA, if you go into MATLAB and use their pre-built GA, they should have these sort of set to default values that work for a lot of functions really well, but then they should give you opportunities to change these. And like I said, you can change numbers like simply your population size, or most sophisticated GAs give the ability to pass in function handles where you can, they give you a spec for a function, like your function is going to be passed in the current population, it's going to be passed in some other details about it, and then you can implement, to your heart's content, your own crossover operator that is like your own way of implementing mixtures of individuals. And we're going to talk about different default crossover operators today. We'll make that clear. You can implement your own mutation operator, and we'll talk about the sort of default mutation operators, your own selection operator, and we'll sort of talk about the default ones. So these are all very flexible, which allow you to really reshape the GA, but the general logic is always kind of the same there. So that's our overall structure about any GA. Any questions on that? So today we're going to make sure that we all are comfortable with the three basic selection operators, with the different ways to cross over and the different ways to mutate. So we're going to cover these because I know those came up in the muddiest points, but that is the general structure. So questions about the general structure? Yeah. Fitness scaling is this, if you want an opportunity to sort of I'll say transform, reshape objective function. So it might be that when you score your individuals with the actual optimization objectives, when you get their observable traits, that coming right out of the optimization objectives, there will be huge differences among certain ones, but maybe not as many differences among others. That might be okay for you, but it might be that just because the optimization objective comes out and it scores a 5 million for one genotype and 500 for another genotype, it might be that all of the genotypes, basically cluster from 1 to 500, except for this one that is like way up at 5 million. Now, if you happen to hit that genotype with certain selection operators like fitness proportionate selection, that genotype is just going to dominate the next generation. And so what fitness scaling does is it allows you to rescale that, and it allows you to sort of replace those raw optimization scores with whatever the actual fitness consequences that you want. And that could be as simple as, like, when we talk about selection operators, ranking selection is an extreme version of this, where you completely replace the cardinal scale that comes out of your optimization objectives with an ordinal ranking. And you basically use the ranking as the fitness. But you can compress these in different ways. Just like sigmoidal functions, activation functions and things like that, they can, if you've got a bunch of or like logistic regression, in statistics and things like that. If you've got one scale that goes from negative infinity to infinity, but you really want to squeeze everybody from 0 to 1, then you can plop that in here and then do your selection on that. And that might change the distribution of individuals in the next population. Yeah, right. I mean, that would be one thing that you could put in here. This is just a way in which you get more, if your selection operator does not give you direct control of this selective pressure that we've been talking about, and we'll talk more about, then fitness scaling is that additional lever to say, all right, well, if I don't have that parameter, like in ranking selection and in tournament selection, there are direct ways that I can play with the selective pressure. But in fitness proportionate selection, there's not as much control over selective pressure. I could add like an exponent. So it'd be like, instead of fitness divided by sum of fitness, it could be fitness to the n divided by sum of fitness to the n. And that would give me a scaling. But that's basically, I just could push that back here, make it more generic. Okay. Any other questions about the general structure? All right, great. So now there is no one GA now. There are a bunch of different approaches. If you, again, download PyGAD for Python, or if you look at GA in MATLAB, or if you were to look in the documentation of genetic algorithms that are implemented in other numerical optimization tools that are in industry, you'd find that they all implement things slightly differently. In the book, we read about one where the population, so this is the GA from the book. I'll put this on this side. So I'll just, to give you an idea of where I'm going with this, I'll just put a little dividing line. So then I'll put the GA from MATLAB over here, or MathWorks, MATLAB. So in the book, they said we're going to structure the population. So this is, population, so that we have, this is the whole population here. And we're going to have a set of our parents and M minus R. I actually don't even want to call them our parents. I like to call them our clonal offspring and M minus R sexually produced offspring. And so the thing that determines the so you set the R hyperparameter, and then there's going to be a subset of these R, which I'll just kind of draw a little this here. It's saying we'll basically call it PCR. So there's some fraction of the R parents that are selected to become the breeding pool. So these become the breeders, which then produce the M minus R offspring. And so basically we have here, this is just probabilistic selection for high fitness clones. And then over here, we've got crossover, which again happens with probability PC from our parents, and mutation. And that's going to happen with some probability PM for each gene of the produced offspring. And so you can kind of think of this as asexual reproduction, and you can think of this as sexual reproduction and then with mutation. And so there are no elites in the book's solution, or in the book's approach to the GA. There are no elites, but they don't mutate these ones here. So they are trying to, as much as possible. There is still, because this is probabilistic selection, so there are high fitness individuals that, due to bad luck in the selection operator, may not get an asexual clone. And that is basically drift right there. So just there's bad luck that may happen, that even though you are the best fitness function, it just happens to be that you had a bad day and the random number generator just wasn't happy with you. And even though you had a huge fitness advantage over the rest, it just happened to be that the dart didn't land on that giant slice of pie, and it landed on one of the other slices of pie instead. And so you didn't make it into there. But if you did make it into there, we're not going to mutate you. So that's sort of the book's way of having something that's almost elitism. So we're going to try to maintain these high fitness individuals as much as possible, but with the possibility that we might lose some. And then of those that we keep, we're going to take a small slice of them, create a breeding population, and then this small slice here that is in our breeding population, we're going to randomly draw from them to create breeding pairs. Those breeding pairs will then combine over crossover to create two individuals that will go into this pool here. Each one of those individuals will get mutated, and that's how we'll fill out the rest of them. And we're going to talk about crossover specifically in just a second. Right now, I'm just saying this is what they had in the book. That's one way to structure these populations. Now, alternatively, if we look at, say, how MATLAB does it, they've made different choices. They do have elitism, and they've sort of spread out crossover and mutation differently. So when MATLAB put their GA together, then their population looks like this. So there's three groups in MATLAB's case. There are your E elites, and then there are of the rest of the population that's left over. So these, and I guess I should write this out somewhere. Let me make sure I'm not going to get in the way. So these are our top E. These are top E elites, and they are just copied directly into the next population. Their fitness, you're saying, I want to take the top 10% of the individuals, for example. I think that's their default is 10% of your population size, and just and deterministically, so no drift options here. If they have a high fitness value, we're keeping them. And so we keep them there, no drift at all, because that's totally deterministic. So I'll just mention that, just deterministic. So because of that, no drift. Now you could argue there's a little bit of drift, because there you may not get into that. Like if there's only two spots for elites and your fitness is pretty much almost just perfect with, really close to that second spot, the one who got the second, then you're not going to get selected for that. But still the top individuals will be in there. So it is a much it definitely is a strong hedge against drift. So then the other sections, the way they come out is like of the rest that are remaining, then they're either so we're going to select parents. So we've got I'll say we've got these M minus E spots left over, and we're going to draw them drawn um, from our parents. So there, you define in MATLAB a selection operator, which you say, we're going to have these many elites and we're going to have this many parents. And those parents are this going to draw from including the elites can make it into the parent pool as well. So we're going to use some selection operator, proportionate, fitness proportionate selection, ranking selection, tournament selection. You have your choices. You can put your own in, but then that will generate our parents into this sort of parent pool. And then those our parents then they have an opportunity to contribute to the rest of these M minus E spots. And they can do that in one of two ways. There are going to be a certain number of spots. So there's going to be M minus E times PC spots. These are the spots reserved for crossover. So these are your sexual offspring. So that's your sexual offspring. And then the rest of these spots, this is your M minus E times 1 minus PC. So the ones that aren't in the crossover fraction. And these are the ones that are your mutation or asexual offspring. So for MATLAB, the crossover operator doesn't get touched by mutation. It just takes two parents in, produces two offspring out, and then it leaves them alone. Then for the other half of these slots, it looks up at those R parents, draws from them randomly, and then mutates those. So it's got the clones that are getting mutated, you've got poor copying in the clones here. So you've got like perfect clones here. You've got clones that are noisy here. And then you've got the sexual recombinations that have no noise. And that's how MATLAB decides up its population. Yeah. Well, I mean, that's anything that has the opportunity for sort of drift. So that and this here, I'll just highlight that selection operator happens here. So this is our selection operator. and then, drift possible. So there are in both of these, there at least are some possibilities where a low fitness solution can still manage to make it into the next generation. And why might that be good? Well, your fitness objective in the GA, we assume the fitness objective does not change during the operation. You could imagine that extensions of the GA that actually allow for, it's sort of like a combination of genetic algorithm and reinforcement learning, where the optimization objective might actually change as like the GA is deployed. But in the standard GA, we don't assume the optimization objective changes. But it might be that there are traits that, or there are, I'll say actually, maybe push it down. There are alleles in your low fitness solutions that if they were recombined with other solutions later, that might actually make for a more interesting individual. So there might be sort of individuals that there's these kind of punctuated equilibria where they're like their alleles have changed, but you need like 6 of those alleles to change for them to actually like to switch into a high fitness solution, but only 5 of them have changed right now. So if they're still functional, maybe we keep them around, and then maybe there'll be some future mutation or some future recombination, which then will cause them to suddenly leapfrog and move through this so-called adaptive valley, where even though they've moved down the fitness hill, then there's something that happens that will cause them to move up a fitness hill later if they just got like the one mutation. Elites are deterministic, but the R parents that populate these come from a selection operator that's probabilistic. So it is still possible that you get into the R parent pool if you're a low fitness solution. It's just unlikely, but it's possible. Other questions? All right, so something else that we'll talk a little bit about when we get to distributed optimization approaches, but MATLAB also opens the door for parallel processing as well. So I'll say plus parallel processing, which allows you to do independent GAs with migration. So I mentioned to you that the 4 forces of evolution are mutation, natural selection, genetic drift, and migration. And migration seems like one that it's important for natural systems, but it doesn't seem like one that might be that important for these artificial systems. But it turns out that if you allow GAs to run separate from each other in the little boxes that don't exchange individuals very often, there's a possibility, for example, that the drift that happens in one box will be different than the drift that happens in another box. It'll be different than the drift that happens in another box. And so you end up getting a different introduction of solutions due to random drift in these different isolated populations. So then if you periodically, after many, many generations, then swap some individuals in between those boxes, it is a new way to introduce novelty. And so MATLAB allows you to say, well, if you've got, 30 threads that are available to me, then maybe we'll we'll create little subpopulations, and then we can then tune how often we swap from in between those subpopulations. So that's another option that MATLAB gives you that wasn't talked about in the books model. All right, so that's, like I said, different GAs, but roughly the same. They all follow basically this structure, but they just might structure the population a little bit differently. These, is there going to be a huge difference between this one and this one? Probably not. This one maybe has a little bit more flexibility than this one. This one gives you maybe a little bit more control over things like drift and so on. So maybe this is a little bit more generic, but it's probably marginal difference in performance between these ones here. So when you're building your own GA, you have a lot of freedom as well. You're basically just shooting for something that follows roughly this structure. You're not shooting for exactly this population or this population. Okay, great. So how do we, there's selection operators that go into these R parents. So how do we do this selection? So our selection operators, Let's make sure that we all understand. And there are three main types, but you can certainly come up with your own and try them out. But these are sort of the ones that have been the most popular over time. So fitness proportionate selection will be our one type, and then maybe I'll try to put these for better or for worse on one page here. Ranking selection. I'm gonna just hedge and not quite put that line up there. And then tournament selection. All right, so fitness proportionate selection is kind of if you come from a stochastic simulation background, then or random sampling background, it's kind of the most natural. It's that you've got these things that already have got these fitness functions. It's actually sort of similar to softmax if you think about it. If you're a neural networks person. So basic idea here is, well, I've got these things that are already ranked by fitness, and some fitness is stronger than others. So let me just draw in proportion to their fitness. So the idea here is the probability that individual K gets into the into the parents R is going to be equal to the fitness of individuals K's genotype divided by the sum of the fitness of from J is 1 to M of all of the other individuals, including that individual. So that's fitness proportionate selection. So you can implement this by basically creating a number line from 0 to 1, where you add up all of these probabilities. So this little region has a width of P1. The next region is going to have a width of P2. This will go on till eventually you have a region with a width of PM at the end. And then if you draw a random number that is distributed between 0 and 1, it will then go to one of these regions. And if it goes to that region, then that would tell you to put 2 in parents. So if these regions are very, very large, then there's a good chance that every time you draw a random number, you're going to put that one into the parent pool. If these regions are very small, there's always a possibility of it, but it's less likely. So that is how fitness proportionate selection works. We can tune selective pressure by fitness scaling, by scaling fitness functions. So basically replacing f of x of k, just replace that with some scaling function of f of x of k. And so that allows you to squeeze and stretch and do whatever you want to do with that. So maybe certain regions of the fitness space, of your objective space, you want to stretch out. Certain regions you want to squish. It's up to you, how you want to do it, or you just want to compress things so that very large differences in fitness don't have that of a large influence on the probability. So fitness proportion selection. Questions about that? Yep. So if you had so the idea here, so let's say if I were to go fitness scale, I'll go back to that page, but if I were to just focus on fitness scaling, if you have an optimization objective that looks like this, for example. So this is my optimization objective. And it might have these large peaks and these low areas here. And so if you've got individuals that are peppered, I've got an individual here that maps to this fitness. I've got another individual here, another individual here, and so on. Then if I directly map this to fitness, then this individual, if I do proportionate selection, will have a huge share of the pie. So if I were to, I'll use maybe a different color. So if I were to map this directly to fitness, so I've got that, that 0 to 1 scale, then this individual might have a giant portion. So this might be P1. So that one goes to P1. And then these individuals, if they're a lot lower, they might each only map to these really small slices of the pie, those small regions. So you might be okay with that. You might say, that solution right there, this is so much better. I am okay if all of the parents in my next generation look like this one. That might be fine with me. But it might be that you say, well, actually, this one's not that bad, and this one's not that bad. And I don't want, I mean, the problem with like if everybody looking like this in the next generation, then that would mean that in the next generation, I might have this same fitness function, but I'm now going to have all of the replacements were going to be pretty much up here. So this is like the result of high selective pressure. So it's going to suck all of these here that are all lower fitness up into that region. And that might be okay with me. If my fitness objective doesn't have a lot of tall peaks, then I might say this is a great way to find the one peak and climb it. But if I do have multiple high peaks and I want to make sure that I've got coverage so that eventually there's a chance of finding that second peak, then I might not want to immediately suck everyone up to the first high fitness peak that I find. So if I do this fitness scaling, so if I then scale, then what I can do is it effectively creates a new set of probabilities so that maybe P1 is still larger than the rest, but it's not that much larger. So this is large, but not very large. And then that helps then maintain diversity longer. So that if down here I've got that same function, then I might get several up here, but I'm still going to get some that are distributed around the rest of it. Does that make sense? Okay. Other questions about this? Yeah. Well, there are various things that affect selective pressure in real life. And so really what we're trying to do is find, so in real life, selective pressure, I mean, the biggest thing that affects selective pressure is population size. And so small population sizes, there's going to be a huge effect of drift. Large population sizes, drift won't be as important, so selection will be more important. Selective pressure in real life is often something that if there's a trait that provides a huge advantage or huge disadvantage, there's no like force or entity that will come down and suddenly mitigate the effects of that. If you have a trait that'll kill you, it's like if you happen to get this particular allele, you won't survive past 10 years old or something like that. There's going to be a high selective pressure to wipe that allele from the population because those with that allele have such low fitness, they don't survive to reproduction, that it is unlikely for that to be passed on. And so that is an example. So there will be selective pressure, there'll be high selection for or against certain traits. And that's just sort of the way of the world. So one way that, if you move into an environment that, you know, phenotype is all gene by environment. So you might have a certain allele that is really deleterious if you live at one altitude. But if you live in another altitude, like if I go to high altitude air, then that thing that, really at low altitudes is causing a problem, I don't even notice it. I operate like everybody else. So that's a way that you can kind of reduce, so there will be less selective pressure against that allele at the high altitude air than the low altitude air. So that's kind of how it happens in nature. This is our way of creating unnatural environments that allow us to create a selective landscape that selects for the things we actually care about, or just makes the search more efficient. And what I hope this also emphasizes is there's a fiction that nature is always going to produce optimal solutions. If there's a high selective pressure like that, really deleterious allele, if one more mutation might turn it into something that gives people superpowers. But if you can't survive, again, this term that I'll introduce later, the adaptive valley, if you have to go down a fitness slope in order to discover a new fitness slope to go up, then getting across those fitness valleys, those adaptive valleys, is really difficult. And that's been a major problem in evolutionary biology, is trying to figure out how this has happened, because it does happen, because just intuitively it doesn't make sense why nature would allow for downward motion, significant downward motion, to permit upward motion later. Yeah. Hmm. Hmm. Well, right. So if someone has a selective advantage, so you do have a superpower, it's only a superpower in nature's perspective if it gives you a much higher probability of reproducing, or a much higher fecundity. So if you reproduce more. So that's how nature does it. Is it like, if there's a new, there's a bunch of animals in a forest, it's hard to find food. Suddenly, all the rest of the animals have really poor vision, and then suddenly there is a variant of an allele that anyone in this line that carries this allele has just perfect vision, and now they could find all the seeds. Well, then those individuals are going to find all the seeds in the forest, which is not only going to give them an advantage, but um, in the presence of the others, those others now suddenly can't find seeds. And so now actually the two of them work together. There's this sort of frequency component there that is going to then push those that don't have that allele are going to then probably not be selected to continue. And those that have that will be selected, and that's how that will spread. Well, the diversity is a big part. Like anything that maintains diversity provides a way to cross it. And so this adaptive value. So this is something that which we'll touch on later, but I'll just mention, since I've talked about it a couple of times now, adaptive valley. So in this case, for those that need a picture, this is a fitness landscape. So this is fitness F, and these are genotypes. Um, and so the idea here is you've got, say, something that looks like this. And so if you happen to be a population that lives over here, so this is where your population lives, then There, it's going to be really hard to have any motion in this direction. And so that's what we mean by crossing this adaptive valley, because this individual is already producing a lot fewer offspring than this individual. If this individual moves down, then its offspring, if it produces offspring which happen to be down here, that offspring will produce even fewer. And so this goes on more and more. And so the idea here is that there's no obvious process for how you're going to get, if you can make only incremental changes to these genotypes, then in order for you to get from here to here, there's going to be intermediate architectures in between, which are going to be really low fitness, which will be outcompeted by these others. If this valley isn't too terrible, I mean, this fitness could be go all the way to zero. So it might be that you have produced zero offspring if you happen to land at the bottom of this valley. But theoretically speaking, if the reason why this thing stops here is just due to that, like if there's sort of a niche that's kind of created, um, yeah, I mean, I would just say, throwing more individuals at this may allow you to fill out this hill a little bit more, but it's still going to be very difficult to get over to there. But what people have found is like one theory for how to get over these adaptive valleys, the so-called shifting balance theory, which we'll talk about a little bit more when we get to distributed. But if you have a small population size, then there's going to be a drift barrier, which is basically going to lop off the top of this mountain so that everybody up here has effectively the same fitness. If drift is so high, then it isn't going to matter if you're here or here, because you'll be so likely to be taken out by bad luck that it doesn't matter if you're a really high fitness individual or a low fitness individual. So genetic drift for small populations can flatten these peaks and make these valleys not quite as deep. So theoretically speaking, if you have a bunch of isolated populations that are all relatively small, then one possibility is that you reduce the selective pressure by sort of increasing drift in each subpopulation, which will allow some individuals to climb down. In other words, there won't be a competitive disadvantage. If the guy that's got the superpowers is just as likely to get hit by a meteor as you, it doesn't matter if they can fly, because you get hit by a meteor when you fly just as well as when you're on the ground, and it kills you just the same. So if you're just sound bombarded by bad luck, by these meteors that don't care what your traits are, then at that point, it gives the underdog an advantage. And that underdog now can survive for longer because those with the fancy traits are now getting knocked out of the population. And so those underdogs stick around for longer, and they might be able to produce offspring that have enough variation that climbs this adaptive valley. And then once they're over here, especially if this is a much higher peak, then they can start climbing it and then discover these new solutions over here. So that's one example of how you can climb adaptive valleys or get over adaptive valleys. But simply dumping more population, that might help fill this out a little bit. But if this is really, really deep, then there's still going to be an advantage. Like it's still going to be, it's not going to be trivial to cross this valley. Okay, it was a little bit of just a diversion, but it was good. Any other questions? Okay, so fitness proportionate selection. So another way that we can deal with the selective pressure is ranking selection. So ranking selection is we go basically from cardinal to ordinal. We throw out the actual giant differences between our different fitness objectives. So the differences between trade doesn't matter at this point. Again, if you think of an optimization objective as like evaluating how tall the genotype is, then here, if you're 6 foot tall versus 4 foot tall versus 3 foot tall, the absolute difference between your heights matters. And ranking selection all it matters is whether you're the tallest in the room or the shortest in the room. So it replaces your fitness with your rank, with your order. So there are different ways to implement this. One way to implement this is just to assign fitness by rank. And so, you can say like, the best gets, some fitness. Next best gets fitness minus Z. Next best gets fitness minus 2Z, and so on. So that's one way you just assign the fitness. So we'll say this is the highest fitness. And so by adjusting this Z, this parameter here tunes selective pressure. The alternative way to do it is to be, and this is basically the same thing, but reproduce by rank into an intermediate pool, and then draw you randomly from the pool. And so here you're basically doing the same thing where we're saying that, but how many offspring you contribute to this pool is basically what's going on up here. So you say, all right, you're the top ranking solution. We're going to give you 100 offspring in the pool. You're the next one. We're going to give you 80 offspring in the pool, and so on and so forth. And then you get this giant pool of offspring, huge numbers. And they say, we need to get our parents from this. Well, then we're just going to draw our parents from this pool, and we'll draw them randomly. And then that will then populate our parents, and we'll throw out everybody else. So this is random. So you do have an opportunity for drift, but it's much more controlled than fitness proportionate selection because it's based entirely on rank and not the raw fitness objectives. But it's basically, you call this fitness proportionate selection. This is like rank proportionate selection. It's not very different. So questions about that? And then the last kind of one that is sort of fundamentally different, tournament selection. And in this case, you select Y. So this is another parameter, candidates from M. And then you pick the best of the Y. And this could be done deterministically. You could just compare their fitness and say, you have a better fitness, always. I'm going to always pick the best one there. The randomness entered into the select the Y from M. That is done at random. So you just randomly select two individuals. So Y, in this case, we'll say Y is often equal to 2. So you select 2 from random, you say which one of you are better, okay, that's the one, I'll take it. And then you put them back. So, and then you redo that. So repeat R times with replacement. And then here, tune pressure with Y. So Y equals M has high pressure, high selective pressure, and then Y equals 2 has low selective pressure. So hopefully that makes sense. The idea being that if you set Y equals to M, then every one of these choices, you're just going to pick the top solution. So you're just going to get R copies of the top solution. But if y is equal to 2, even if you're at the bottom of the ranking, there's or close to the bottom of the ranking, there's an opportunity that you'll go into tournament with someone who is lower than you. So you don't always have to compete with the top. Here and here, there's always some competition with the top. Here, there's at least, there's a significant opportunity that you won't compete with the top individual. You might compete with individuals higher than you, and if you're at the bottom, it's very likely they'll be higher than you. But if you're kind of in the middle, there's a good chance you're going to win a lot of these tournaments. So that's how we tune selective pressure. Questions about that? And so getting the ability to tune selective pressure helps you maintain diversity for longer, which will hopefully let you search more of the space. Okay. All right, so that's the hardest of the operators we have to deal with. So let's cover the others then. So then the other issues, crossover. So in nature, during meiosis, during whatever prophase 1, I guess, or something like that, don't quote me on that, there's a process where homologous chromosomes, so if you get the two of the same chromosome come together, and there's this little thing called a centromere, which sort of holds them together, makes them kind of form like an X, and that kind of organizes them. And then at certain points along, and they're usually not close to the centromere, so they tend to be closer to the ends of the chromosomes, and they tend to be on the longer side of this X that's formed. Then you get these random points where parts of the chromosomes will come together, and then they will swap genes on either side of that constriction where they come together. And this often happens in one point, again, often on the longer side of this centromere. It's often not close to the centromere, but it can happen multiple times, 2 times, 3 times, 4 times. The more times, it's less likely, but it at least happens once. And that allows for some of the parent A's alleles to be combined with some of parent B's alleles. And that's what we're mimicking in the crossover operator. So the idea here is that so at this point, because I'm trying to be generic, we've already selected the parents for crossover. So let's assume that there were our parents, and we have selected PCR slice for our breeders. So, I'll just call these breeders. So we're going to select 2 breeders at random, which I might call X1 and X2. And then so we're going to line up their chromosomes next to each other. So that might look like this. Maybe that's a little bigger than I need it to be. Might look like this. So this is X1 and this is X2. And so in 1-point crossover, so I'll push the we then pick a random gene that serves as a boundary between the things that'll be crossed over and the things that won't. So we just choose this at random. So we're going to pick a spot, let's say right here. So pick at random. And this is just a boundary between genes. And then this individual, I'll just patch as all of their alleles, and this individual, I'll put little dots. And then we're then going to create 2 offspring, which I'll call Y1 and Y2. So these are our two offspring. And they're going to match. So this is also the same spot. They're going to match their parents on the left, and they are going to swap their parents on the right. So on the left here, we'll have all the hatching. So all of these genes will come from parent one and parent two, but then up here, we have the switch. So basically swap after crossover point. All right, so that is 1 point crossover. Questions about that? Does not change the distribution of the alleles, but it creates new combinations of alleles. You can then have 2 point crossover, which will look very similar to that. So I'll just draw a little sketch of that 2 point crossover. Where basically, so we've got our X1 and X2, we pick two boundaries at random. So X1, again, I'll hatch. Then I'll do little circles for X2. And then after the crossover, we create two offspring, same boundaries. So then those boundaries go here. And then so we'll say that, and this will produce Y1 and Y2. And so we'll say they match here, and then they swap in the middle, and then they match at the end. And then you can imagine that you could have multi-point crossover. So you could have 3, 4, 5, etc. And you can tune how many of these crossover points you want in a standard genetic algorithm. You can also have uniform crossover. And so here you basically define a probability. So there's a probability that any gene will cross over. So in this particular case, if you line up these two genotypes next to each other, then we've got all of our genes here. You basically put a boundary at each one of these genes. So these are my N genes. And then there is a probability. So we can just ask, does this cross over? Does this cross over? So you can basically say there's probability of crossover or of swapping at each gene. So in a uniform crossover, you just march down down the chromosome, and you say, and you flip a coin, and let's say there's a 50% crossover probability. Then we say, does it come up heads here? Okay, swap. And so Y1 and Y2 will be swapped. Does it come up heads here? No? All right, we'll leave those alone. Come up heads here, low, nope, leave them alone. Here, swap, and so on. So you just, for every gene, you flip a coin to figure out if you do crossover, and you can tune that probability, and that gives you a uniform crossover probability. And then alternatively, you can replace this complex crossover with a linear combination. So an alternative linear combination, or I should say convex, often combination. What do I mean by that? It basically says that I can say draw alpha from a uniform distribution between 0 and 1. And then y1 will be alpha times x1, assuming these are all numeric, plus 1 minus alpha times x2. And then y2 might be then 1 minus alpha times x1 plus alpha times x2. So this is a way of, so you don't have to line things up in some data structure and swap and reassemble. Then this is a way which captures a lot of what crossover is doing anyway, is that basically these are two random interpolations between parents. And you can also do extrapolations for other values of alpha. So, I mean, that's really what sex does, is it creates a blending of the two parents. Well, one way we can just capture that is if they're all numerical genes, then rather than just swapping, then I can blend by just mixing these things directly with those things. And so that's another way to do that. So questions about that? Those are some common ways to do crossover. Okay. And then let's just then at least bring it up so that we all see it. And then I'll just make sure I'll probably start here on Thursday just to make sure that we've got mutation down. But then the last one left is the mutation operators. And there's in mutation, the simplest thing to do is just you've got some probability PM of a gene mutating. And so for every individual that is in your mutation pool, you just walk down their genes and you flip a coin to see if that gene should be mutated. And then there's a bunch of different ways you can mutate it. So, uniform mutation is you basically just replace the gene with a random gene from the set of possible alleles that are associated with it. So uniform mutation is like if I've got my individual J and this is gene I, then I'm just going to set it equal to or draw it from, so this is a little bit of an abusive notation, a uniform distribution where this is the full range. So uniform draw from full range of alleles for gene I on individual J. Now, that may be too strong for you. And so the other major way to do this, and then we'll talk about the other ones next time, is alternatively, we can just add some noise. And so in that case, you can do something like xji is equal to xji plus some uniform noise that has a particular width. And so this is like, zero mean noise between W or minus W and W. And then you could choose different distributions for this as well. So these are, this is the sort of the most kind of uniform way where you just blow away the allele, or you just perturb the allele by a little bit. Now, what we'll close with then starting on Thursday is that both of these can be tuned over time, or this can basically be tuned over time, where you can initially make it act like this, where it just dislodges the entire gene. But then as you march on and get later and later in the algorithm, this width gets smaller and smaller so that it just makes fine-tuning arrangements until when you end, you're hardly adding any noise whatsoever. So that is going to be very similar to simulated annealing when we get to that, when we get out of the genetic methods. But that's all I want to got for you today. I don't want to keep you any longer, so I'll see you Thursday.